Ladies and gentlemen, I want to present you information based on our results, mainly on COVID-19, which is now called COVID-19, long COVID-19, or after COVID, or after COVID syndrome. First of all, I want to show you some possible mechanism of this after COVID syndrome, COVID complications, and examples of some cross-reactivity of bacterial antigens with human tissue will be introduction to this presentation. So most often molecular mimicry is a mechanism of pathogenicity. Participation of bacterial salicylic acid is basis of very often is basis of bacterial complications. For example, gram-negative bacteria have a cell wall which possesses outer membrane endotoxins, lipopolysaccharides, and the most external part is polysaccharide in this bacterial antigen. Molecular mimicry is a structural, serological, and functional homology of between microorganism and its host. Auto-immunization takes place during the infection, and humoral and cellular response is directed to auto-antigens, antigens which are very similar to human tissue. For example, coli K1 or Neisseria meningitis B have such a linear polysaccharide composed of salicylic acid. This same polysaccharide is human brain tissue. So polysalicylic acid in higher organisms together with human tissues is in E. coli, in Neisseria meningitis group B, and in neuronal cells in embryonal rat kidney, and in also very often is in neuroendocrine tumors. So very good example is of similarity between bacterial antigens and human tissue is Campylobacter jejuni antigens. It's part of lipopolysaccharide, surface located antigen, and during prolonged infection with Campylobacter jejuni, this antigen, which is identical to gangliocytes, human gangliocytes, can be autoimmunity with this bacteria, and antibodies are induced for these bacterial antigens. Another antigens of Campylobacter is O1, O2, O4 antigens, and several. Also in Helicobacter pylori is also example of antigens similar to human Lewis X, Lewis Y antigens. So these antigens are identical to human gangliocytes and glycoconjugates, which can induce antibodies, autoantibodies in human tissue after the infection. Another example of similarity is E. coli O2456, which have also sialic acid in their structure, and we have immunohistochemical evidences of cross-reactivity of such antibodies against these structures, which could be identified in such cross-reactivity in nerve with nerve ganglia and in neuroblastoma. So we don't know yet the direct relation with that autoantibodies are formed, but the cross-reactivity we have antibodies against bacterial antigens, this one, with human tissue. So it is should be studied more. What about viral proteins? We have here. I found interesting results of studies that group of people, young people, have antibodies against S1 protein of SARS-CoV-2 virus after vaccination. For steps of vaccination, these antibodies are growing in serum, and in older people, these also are growing. The content of this antibodies is growing. Interestingly, is that people which were not immunized also have growing the level of these antibodies in younger and older people. So we have an example of induction of antibodies against virus. But what about the symptoms of so-called long COVID or after COVID complication? Long COVID symptoms, which is now is problem that COVID itself is milder clinical symptoms, but after this COVID, for few weeks, complications are when exists extreme fatigue, shortness of breath, cough, hoarse voice, problems with memory and concentration, problems with sleeping, and changes in mood, depression or stress, muscle pain, and other symptoms. So long COVID can also show up as new chronic conditions, such as heart disease, diabetes caused by this virus infection, kidney disease, and what is important, neurological conditions. This is most often symptom of this long COVID or after COVID syndrome. When we have SARS-CoV-2 virus proteins, we can see that the surface of this virus is composed of S protein, spike protein, E, it means envelope protein, membrane glycoprotein. These are glycoproteins. It means that they contain sugar part. And N, nucleocapsid protein, which is inside, is without carbohydrate domain, but appears that it is it happens quite often in virus that most immunogenic proteins, components of virus, are not the surface antigens, but is nucleocapsid proteins. So it's in this case also is that S protein, for example, it's overloaded with carbohydrates, and this receptor binding domain is here on without carbohydrate. This carbohydrates contains mannose,ylation, fucosylation, cellulation. So this makes that such a protein is here protected to immune system, and most immunogenic is just this R receptor binding domain. Regarding nucleoprotein and protein, let me come back to this picture. These proteins are studied for vaccine as a component vaccine, but this nucleoprotein and protein was not considered as a vaccine component because some publications revealed that antibodies against this protein and protein are increasing the disease conditions. They are pathogenic. These antibodies in patients, COVID patients, they are not safe. So it was not considered for vaccine production or vaccine construction. We can see that when we have electrophoresis picture of SARS-CoV-2 proteins, most important is just most immunogenic is N protein, and S proteins are less immunogenic, less antibodies are visible here in this picture. So main band is against it means that antibodies against this N protein are very high concentration. It was revealed that part of these antibodies are harmful. So our studies revealed that this N protein contains 7 fragments which are identical with 7 human proteins. Cross-reactive proteins with this N protein are identical to 7 human proteins. So let's show these proteins. Ninjurin-1 is a human protein which is transmembrane protein expressed mostly in endothelial and myeloid cells. And what is important is involved in this protein is involved in progressive inflammatory diseases. It's an adhesion molecule which plays a role in programmed necrotic cell death in neuroinflammation. So we see that this protein, human protein, which has identical sequence of amino acids with N protein, is involved in neuroinflammation. The second protein, human protein, MDC1, which have very important functions in human organism is estrogen receptor co-regulator, has important functions in this protein which indicates that potential induction takes place in potential induction of autoantibodies against this protein could be harmful. It means that it's not directly cause neurological symptoms, but such antibodies against this protein may be also harmful. Another protein, human protein, which have fragments identical to N protein, viral N protein, is SURF1 protein, which is involved in mitochondrial translation regulation assembly, intermediate of cytochrome C oxidase complex. So defects in this protein cause several neurological disorder and the other disorder of the peripheral nervous system. So this protein, when could be blocked with autoantibodies, could also lead to these neurological disorders. This fourth human protein with a similar epitope sequence to N protein is acidic cluster sorting protein, so-called, and could be used by virus for immune evasion, multiplication, and pathogenesis. And is also a mutation if this gene of this protein is involved in neurodevelopmental disorder, involved in neuropathology. So we have third one. Another human protein which have sequence similar to protein N is T-GOL-N, which have important functions in this protein could be also harmful when antibodies could be directed to this protein. Another protein is serine protein phosphatase, which also have sequence which could be identical to N protein. And the last one, DNA homolog subfamily C member 3, which is which also autoantibodies, when induced to this protein, can be involved in induction of ataxia, combined cerebral and peripheral, with hearing loss and diabetes mellitus and influenza. So this is very important protein can be involved in neurological disease. So concluding, this highly immunogenic nucleocapsid N protein of SARS-CoV-2 virus stimulates the production of antibodies during COVID-19, with recognized sequences in human proteins, at least 4 of which are responsible for neurological diseases. So our hypothesis is that induction of autoantibodies may be the cause of long COVID symptoms. So another point is that there is a need for COVID-19 prevention, production and use of a safe and effective vaccine to prevent COVID-19 and long COVID. So when we have vaccine against COVID-19, we can also prevent long COVID. How is work on our vaccine? In use are several vaccines against COVID, attenuated, inactivated vaccine, DNA or protein subunits, virus-like particles, virus vector and mRNA vaccines. In use is this vaccine based on mRNA. In China also is another one. Protein subunit is not based on nucleic acids, will be soon in many different countries. So our project is on this step. We have prepared our vaccine based on subunit on M protein, E protein, and S protein for studies that phase 1 and phase 2 studies, and we consider also fragment of N protein which is not cross-reacting with human proteins. Currently, many laboratories are working on vaccines. Some of them are under clinical trials, which indicate the most intensive development of vaccine research and testing in history. This picture shows that some correlations between different companies are involved in vaccine commercialization. Here I present my group, which is involved in elaboration of our vaccine. Thank you very much.